Assalamualaikum and what's up guys? Lokman is here. Today I represented my group to present our lab report which is enzyme. Before we go further, we must know what is enzyme. Enzymes are built of protein folded into complicated shape. The chemical reaction that keeps us alive, our metabolism rely on the work that enzymes carry out. Enzymes speed up chemical reaction. A substrate binds to the active site of an enzyme and is converted into products. Once the product leaves the active site, the enzyme is ready to attach a new substrate and repeat the process. There are many factors that affecting enzyme. First, enzyme concentration. Second, substrate concentration. Third, effect of temperature. Fourth, effect of pH, and lastly, effect of activators. For the first factors, enzyme concentration. As the enzyme concentration increases, velocity of reaction proportionately increases. For the second factors, substrate concentration. The rate of enzymatic reaction increases as the substrate concentration increases until a limiting rate is reached. Further, increase in substrate concentration produces no significant change in the reaction rate because the enzyme molecules are saturated with substrate. For the third factors, effect of temperature. Enzyme activity gradually lowers as the temperature rises more than the optimum temperature until it reaches a certain temperature at which the enzyme activity stops completely due to the change of its natural composition. For the fourth factors, effect of pH. Each enzyme has the pH value that it works at with maximum efficiency called the optimum pH. If pH is lower or higher than the optimum pH, the enzyme activity decreases until it stops working. For the last factors, effect of activators. Some of the enzymes require certain inorganic metallic cation for their optimum activity. Rarely, anions are also needed for enzyme activity. Now, I would like to present our result for this experiment. Before that, in this experiment, there are two parts that we need to do. First, salivary amylase activity. Then, kinetic of amylase. Under kinetic of amylase, there are substrate and temperature effect. So, this is our standard reference graph. For the first activity, salivary amylase activity, we collect the saliva among each of us, then we observe and compare before and after do the stress activity. Here, this is the result. This is the result for the before do the stress activity. This is a result after the stress activity. The concentration of substrate we get from the standard reference graph. So the discussion for this activity, we can see that the absorbance for all the students before do the stress activity is decreased after do the stress activity. The amount of absorbance is decreased while the concentration of substrate, starch, also decrease. This is because salivary amylase plays a role in the breakdown of starch into glucose and maltose. Salivary amylase may prove to be a valuable stress marker due to the saliva gland releasing enzyme upon stimulation of the sympathetic nervous system during exercise. So, most substrate, which is starch, has been catalyzed by the salivary amylase during the stress activity. That's why the concentration of substrate after the stress activity is low. Next, for the second part, which is kinetic of amylase. First, we observe the factors that affect enzyme activity, which is substrate concentration. So, this is our result. From the result, we plot the graph 1 over V against 1 over concentration of substrate. The discussion for the substrate concentration: as the concentration of substrate increases, the rate of reaction also increases and will remain constant at the saturation point. Because as the concentration of substrate increases, the enzyme becomes saturated with substrate. The rate of reaction when the enzyme is saturated with the substrate is the maximum rate of reaction, which is Vmax. Then we observe the second factor that affects the enzyme activity, which is temperature. The temperature that we choose or use in this experiment is 25 degrees Celsius, 35 degrees Celsius, and 40 degrees Celsius. This is our result: 25 degrees Celsius, 35 degrees Celsius, and 40 degrees Celsius. From the result, we plot the graph 1 over V against 1 over concentration of substrate. So this is our 1 over Vmax and negative 1 over Km. Discussion for temperature effect using amylase. Temperature is the one of the factor that affect the rate of reaction. The higher the temperature, the higher the rate of reaction, because the collision frequency is increasing. Particles can only react when they collide. If we hit the enzyme, the particles move faster and collide more frequently, which can speed up the rate of reaction. Based on the theory, the optimum temperature for the enzymatic activity of salivary amylase is 32 degrees Celsius to 37 degrees Celsius. The optimum temperature means that the temperature at which the enzyme shows the maximum activity. In this lab, the amylase look for function at 40 degrees Celsius, then 35 degrees Celsius, and 25 degrees Celsius. That's all. Thank you. Bye.